Yes, yes, we can see your screen now.
Yes.
Okay, I think it's now the time to start. So, good afternoon, everybody, and welcome to yet another Mindray Masterclass. Today, we have the session on quality assurance and NABL documentation, which will be taken by Dr. Neeraj Jain, sir. As we know, Dr. Neeraj Jain, sir, is the course director and the president for Medical Education and Learning Point. Sir is also the lead assessor for ISO 1589-2012. Sir is also the principal assessor for NABH. Sir is highly, trained in and has the expertise on Six Sigma Green Belt, ISO 17043 NABL, ISO 13528 NABL. Sir has been given a lot of rewards and accreditations across the different organizations, such as the National IMA Award of Merit in 2014, IMA National President Award in 2014, DMA Annual Award in 2014 and 17. He has been awarded at CMC Vellore. Sir has been Sir's Institute, which is the best institute, has been named as the best institute for health education and training in 2020. So we have among ourselves expert person who has really a very good experience on quality assurance and lab documentation. Sir has been taken various classes on this matter, and sir is very passionate about the quality assurance and how the different labs and giving them solutions, through learnings and teachings. So I would like to welcome Dr. Neeraj Jain, sir, in behalf of all the attendees and the Mindray team. Sir, over to you. Thank you.
Good afternoon, everyone. I think all of you will agree with me. And this is the things which person cannot understand. All of us have only one body and one soul. And both of these cannot exist without each other in this living world. And a lot of progress is happening in the medical world to keep both together. And all of us have realized this. In the last few days, government is working very hard. Doctors and all medical fraternity is working very hard. And in fact, whole humankind all across the globe is working hard to keep both these together. And when we are staying at home, maintaining social distance, we are free. We are also worried what to do, how to pass our time. So we realized we should develop something. We should spend some time in enhancing our knowledge. If you see what is happening in the medicine, nowadays everybody is talking about how we can diagnose this COVID-19 in an unequivocal manner. If we are saying this test is positive, it is not cross-reacting with other SARS or MERS or other influenza virus, and how we can detect it at as early as possible. Same is true for all diseases, not only this corona. We are talking a lot about this corona nowadays, but this is true for everything, whether it is histopathology, cytopathology, or hematology. What should be our diagnostic strategy to diagnose a particular disease? What should be algorithm of treatment? A lot of progress is happening in molecular medicine, and people are talking about immunotherapy in this COVID-19 also. Can we give a patient plasma therapy as a treatment? People are talking about stem cells in various diseases, nanotechnology, personalized medicine. A lot of molecules have already been developed, and we can start targeted therapy based on positivity of a particular molecule. New drugs we are developing, and now everybody has realized the importance of clinical laboratory and a documented quality management system in the laboratory. And you have seen for this COVID-19 also, ICMR has allowed only those laboratories which are accredited as per ISO 15189, because we are concerned about patient safety. What we mean by safety? Safety means patients should get correct report, so that patient is receiving only correct treatment and receiving treatment only when it is required. And a lot of things are happening in our laboratory in particular also. Laboratory automation, we have started this robotics also, consolidation of laboratories. At a particular central test, all referral tests are available. At the periphery, all routine tests are available. Lot of role of IT in the laboratory. Progress is happening in the molecular diagnostics like DNA microarray, proteomics, pharmacogenetics. We are talking a lot about point of care testing, whether it is related to diagnosis of various infections, arterial blood gas, or a simple test like glucose monitoring. Nowadays, we have this point of care testing for coagulation studies also. Previously, we used to think these imaging services are separate. Our laboratory services are separate, although these are separate. But now people have started developing the concept of integrated medicine. They are correlating these lab finding with imaging, with images, and providing clinician a final diagnosis. In pre-analytical, we are using this barcoding to avoid pre-analytical errors. For transport, pneumatic chute is there. And to ensure quality and to assure somebody else about the quality, this accreditation of laboratory is a very good thing. What is the expectation of a patient or a doctor from us? These laboratory tests shall be available in a laboratory, and all tests required shall be available. There shall be comprehensiveness, and that turnaround time should be as minimum as possible. Whatever reports we are generating in our laboratory, those shall be accurate, and clinician can take decision based on our report. They are thinking our reports are reliable. That means accurate reports are reaching to them within reasonable time frame and in unambiguous manner. And whatever report we are generating, if suppose they want to ask something, we are there to help them. And it is our duty to provide information, whether it is some pre-analytical requirement, how to collect a particular sample, how much sample shall be collected, how frequently a particular test shall be repeated, or if they want to discuss, patient or clinician want to discuss interpretation of a particular test. As a laboratory person, this is our responsibility to help them. And if suppose some patient has some complaint or claim, we should be ready to help them. All of you will agree with me, 70% clinical decisions are based on our reports. Therefore, our report should be error-free. It should be meaningful for clinician so that they can make a correct diagnosis. They can start correct treatment based on our report, and patient is receiving a better care. Sometimes people say, okay, all these things are good, all these things are acceptable, but we are maintaining a reasonable quality, we are maintaining a good quality. This accreditation is required if we are going for some panels, or if it is the requirement of some hospital and we want to start some laboratory there. But quality is for everybody, because every patient, every doctor wants correct report. We cannot say ki we are testing only 25-30 samples daily, therefore these controls are not for us. The CMC or AMC of machine, or the proper maintenance of the machine is not required by us, or calibrators are not required by us. These are mandatory requirements for every laboratory, and it hardly matters whether you are going for accreditation or not, but something cannot be ignored. Now, various terms which we use in our laboratory, what is the meaning of quality assurance? Sometimes we use these terms interchangeably, but what is the meaning of quality assurance? We are telling our clinician, we are telling our patients, you will get correct report within reasonable time frame. We are assuring them about quality. To assure them, we follow some quality requirements in our laboratory, whether it is related to proper documentation, whether it is related to development of various procedures, related to various activities, whether it is related to different phases of testing, pre-analytical, analytical, and post-analytical. We are addressing all these requirements. So for this, we should have a documented quality management system, which is very well implemented in the laboratory. And regularly we are auditing our services and we find out areas where we fail to comply with the requirement and where we can further improve our services. Sometimes people say, our laboratory is a very good laboratory. Why? Because we have a machine which is the only installation in the country or only installation in the city. Nobody else has this machine. But remember one thing, if our pre-examination and post-examination processes are not well controlled and we bring in automation, what all we get is automated mess, and 80 to 85% errors occur during these phases and particularly during pre-examination, and our results are only as good as the quality of sample received in the laboratory. So therefore, it is very important to maintain the quality of specimen and how we are generating our reports in addition to analytical quality. Sometimes people say our laboratory is ISO 9001 certified. We have this certificate. This is related to quality. We don't need any accreditation. Both these things, certification or accreditation, is by third party. First party laboratory, second party your users, and third party which is neither service provider nor user. They are saying your laboratory is fulfilling quality requirement. In certification, only documentation is checked. In accreditation, we check technical competence also in addition to documentation. And in our healthcare system, this technical competence is very important because we don't have any second chance. And we cannot replace anything. If suppose you are going to market, something is not of good quality, the shopkeeper will change the product. But here you don't have any option. What is important? Technical competence is very important. And this accreditation is a good way to demonstrate competence of the laboratory. And with this, you recognize a particular laboratory. Nowadays, what is happening? ICMR said laboratories which are accredited for RT-PCR, for viral detection, only those laboratories will be allowed to perform this COVID testing because they have made an understanding if this laboratory is accredited, that means they have a documented procedure and they have a competence. This is demonstrated competence of the laboratory. And in future, you may find this situation in various other diseases also. You don't know. And other countries, in a time to come, they may say, we will accept only those research papers where testing is being performed in an accredited laboratory. It is better we start developing our system as per this international document, ISO 15189. It is already mandatory in some countries, and many countries may make it mandatory in the future. And when we are going for accreditation or we are working in an accreditation laboratory regularly, there are audits, internal audit or audit by external assessors. This is some sort of stimulant for us to keep this quality system alive. When we follow this ISO 15189 laboratory, definitely there is reduction in errors in the pre-analytical processes because we develop our system we are more confident about our reports. Repeat analysis, redos, these are decreased. When clinicians are getting accurate report within reasonable time frame, they can start treatment early. This also help us in continuous improvement in the laboratory. And accreditation not something which laboratory is best. This is we check compliance with the standard procedures. This laboratory is working with reasonable quality. They are following standard procedures. This is instrumentality, not aim. And this quality system is a never-ending story. This is a journey without any destination. You have to maintain your quality system and regularly try to improve your quality system. Quality system, this is about people, for patients, with people, by us as laboratory professionals, and for people, that is our clinicians. They are taking decision based on our report. With this accreditation, there is standardization in the process, demonstrability of result, and our personnel, they have certain guidelines, how they will develop themselves. Evaluation of supplier, we are getting good quality reagents, what all shall be checked. In this, we document that, and we can communicate our partners, our patients, our doctors, our laboratory professionals in a better way. Although there are some reservations for people. They say, in our college time, we have not learned all these things. Nobody taught us all these things. What is this quality management system? We know our histopathology, we know our metapathology or biochemistry or microbiology. Now it is very difficult to learn these, but learning has no age barrier. You can start learning at any age. This calibration, this is an extra cost for laboratory, control material, extra cost for this validation, so much reagent will be used, and expenses of the accreditation. It is time consuming process. We have to appoint more personnel, and insurance company, other payers will not pay us extra money. But all these things are okay, but still we cannot compromise on the life of patient. Right patient should receive right test at the right time by the right process. This should be our intention, and every laboratory should follow this. Now we have discussed so much about quality. We should work with the quality. But what is the meaning of quality? Quality means conformance to requirement. And what is the requirement? Our all personnel should be qualified. Qualification means educational background plus any training or experience required. If suppose some oncology institute want to appoint one histopathologist, he would like to appoint a person who has degree of MD pathology plus training or experience in some oncology institute. If suppose it is a small hospital, routine hospital, if suppose they want to appoint somebody for histopathology, a MD pathology with 3-year experience may be sufficient for them. So this and suppose you want to appoint somebody for flow cytometry, then you require, in addition to qualification, in addition to education, some training or experience related to flow cytometry, and same is true for your various different activities. Then you should have a system of training so that there is harmonization within the laboratory and everybody is getting opportunity to upgrade their knowledge and skill. Then competence assessment. What is competence? Ability to use acquired knowledge and skill in practical situation. Problem solving skill, you are asking them to test a pre-analyzed sample, observe how they are performing various activities in the laboratory, how a particular technician is cutting section, how the person is staining slide, how a person is maintaining various machines. You see all these things, and if you think there is some compromise, then conduct some training session for them. Then people should get opportunity to upgrade their knowledge and skill through CMEs, seminars, various training programs. Either you can send somebody to other organization, other city, or you can call somebody to conduct sessions for them, and you can develop some in-house system also for all these activities. But remember one thing, keep record of all these activities. For every training, there should be post-training evaluation, whether the people have understood the contents of training or not, because ultimately that knowledge will be used in the generation of reports within your laboratory. So this post-training evaluation, competence evaluation, performance evaluation, regular participation in these conferences, and post-conference evaluation, all these things are important. And our report is only as good as the quality of specimen received. And one very important thing where all of us as a laboratory person face problem, a properly filled test request form, particularly in histopathology, cytopathology, bone marrow examination, and in coagulation studies. But this is important for every test, and you discuss with your clinician, discuss with your management if you're working in the hospital, so that you're receiving a properly filled test request form. Specimen is correct. Correct specimen means patient is identified correctly, and sample is collected in the correct tube, and you are collecting a proper volume, and then sample is mixed properly, and then it is labeled. International requirement is sample shall be labeled after collection in front of patient. Then sample is transported to the laboratory, keeping in mind safety of the person who is carrying those sample, integrity of the sample, and this sample shall be transported within reasonable time frame and at appropriate temperature. When somebody receiving in the laboratory, some authorized person shall receive, and the person should check sample is properly labeled, correct volume is collected, and there is no clot in the coagulation tube or in the EDTA tube. And information on the sample is matching with the information on the test request form. Then that person is signing with date and time on the test request form. Everything should be well documented for any future reference. People should get phlebotomy training, which in addition to collection of sample from the patient shall include all these aspects and how to document various activities related to sample collection. Now, these reagents, these personnel, these reagents, and our training area, these are called resources. These shall be well maintained. We have to address certain quality requirements related to all these things, and we have already discussed personal part. These reagents, when we are receiving in lab, what all we will check. We have to check with the purchase order, whether informations are matching or not. We have to check with challan, whether information, lot number, pack size, all these things are matching or not. In addition, visual inspection, this kit pack is okay, it is not distorted, it is at appropriate temperature, then it shall be stored under defined condition. Now, after addressing these requirements, still we are not sure about the quality of reagent inside, whether it is appropriate for patient testing or not. So how to check that? That is called lab acceptance testing. Suppose it is some stain, Leishman stain, Zn stain, Gram stain, or your various stains related to histopathology, cytopathology, other things. You should have a documented procedure for that. And what is your acceptability criteria? Then actual performance of that acceptance testing, and some authorized person has to approve that for patient testing. You should have record of all these things and proper documentation of all these things. Sometimes we are confused. What is the meaning of document? What is the meaning of record? What is document? Any meaningful information on any suitable medium is called document, whether it is hard copy or your soft copy. And evidence of implementation of some activity is called record. If suppose you are writing a procedure, how you will address IQC in your laboratory, that is your document, that is your procedure, but that is not record, because that is not evidence of some activity. If you are running IQC, you are filling some format, then you are keeping a record of instrument printout, then you are keeping record of LJ charts, corrective action taken, instrument printouts. These are also useful information. And you are maintaining some hard copy or soft copy. These are evidence of implementation of activity, these are record. That way our all records are documents, but all documents are not record. We were discussing this lab acceptance testing for this stain. Suppose for Leishman stain, we are keeping few unstained slides of normal patient, some slides of thalassemia trait, some slides of eosinophilia. And suppose for any new lot, when we are preparing a new lot or we are buying a prepared reagent of new lot, we are first staining these unstained slides. If suppose we are able to see neutrophil granules clearly, able to see eosinophil granules clearly, if we are able to see polychromatophils, platelet color morphology is okay, RBC color is okay, that means it is of good quality. This is of acceptable quality. After checking all these things, we can approve our stain for this peripheral smear. Same is true for your other different type of stains. For AFB, you can keep some positive and negative slides. For new lot, you're checking with positive and negative cases and you're documenting that and somebody is approving. For card test also, you keep in your storage some positive and negative cases. Whenever you are starting a new lot, test with positive and negative. If a positive is coming positive, negative is coming negative with this new lot of reagent, that means this lot is of acceptable quality. You can document that. For quantitative test, your hematology, your biochemistry, whether it is routine biochemistry, hormones, or tumor markers, whatever criteria you are following for your daily approval, suppose you are running controls daily and you are plotting LJ chart, with this new stain also, you run that control. If suppose results are close to your previous results, that means this reagent is of appropriate quality. You can approve that. So that way we can do this lab acceptance testing. In hematology, sometimes we face some problem. This we will discuss at a later stage. Now, this is important for as a laboratory person, for us, as well as this is important for manufacturers and suppliers also. And sometimes we get all these things in the literature. What is the meaning of this? What is diagnostic sensitivity? What is the possibility of getting a test positive if a patient has disease? Diagnostic specificity. What is the possibility of getting a test negative when the patient doesn't have disease? Now, this is from the case perspective. From the test perspective, positive predictive value means if a test is positive, what is the possibility of having that disease? If a test is negative, what is the possibility of not having that disease? And in test utility, we see all these things. In addition, we see cost of test also. We see how much time it will take, how much expertise is required. On that basis, what type of equipment is required. On that basis, we decide about the selection of a particular test. Now we have to decide about a machine. Suppose you are planning to start a new laboratory, or you are already in a lab, you are running a lab, you want to buy a new machine, say hematology analyzer. First, you have to decide what all you are expecting in that machine. First thing, whether you want a desktop model or floor model, what should be the throughput of the machine, linearity of various parameters, then precision of various parameters, carryover, or in addition, certain things like it should have this autoloader, slide dispenser, slide stainer, whatever you are expecting in a particular machine. Then based on your desirable characteristics, company people, they are providing you information of their various models. In this model, you will get these things. In this model, you will get these things. In this model, you will get these things. And these type of information you will get from different manufacturers. Now you have to decide based on your desirable characteristic, which machine is suitable for you. Suppose you want a machine of throughput 100, and company people is saying we have one model of 80 tests per hour, another model 150 tests per hour. One machine is costing you, suppose, 5 lakh rupees. Another machine is costing you 12 lakh rupees. So you have to decide what is your requirement. So now you're matching your desirable characteristic with their specification. And for certain things, when after buying a machine, you're checking, suppose they are saying in our machine for hemoglobin, linearity is up to 25 gram per deciliter and precision is 2%. They may say before buying that machine, you keep your demonstration machine here, we will check what linearity we are getting, how much precision we are getting. And they may find company is claiming 25 gram per DL, and suppose they are getting around 24.5 or 24 or something like that, they will say, okay, performance is this machine is satisfactory. Whatever they are claiming, we have checked and we are happy with this. And after getting machine in your laboratory, you are getting a new machine. In your laboratory, you are checking these things. Okay, they are saying precision is 2%. What is the precision I am getting in my laboratory? So what they claim, that is called performance specification. What you get in your laboratory, that is called performance characteristics. Then we have to understand in instrument comparison, what is linear regression analysis and correlation coefficient. Suppose you have one machine. You're happy with that machine. You're testing hemoglobin of different ranges in this machine from 5 to 25, 5, 10, 15, 20, 25. What is your expectation with the new machine? Your expectation with the new machine is, I shall get similar result. But we expect so many things in our life and we don't get all those things. So if suppose for this, if we are getting 5 here and we are getting 5 in new machine also. If suppose we are getting 10 with the old machine and 10 with the new machine, that means we will have graph at 45 degrees. That is ideal situation. But if suppose with this new machine, if we are drawing different points and we are if we are putting different points and then drawing a line, and the line is almost overlapping with this 45 degree, that means with this new machine, correlation is very good. But if we get this light colored line, like one line parallel to this line, but above it, and there is an intercept. First line was starting with 0 and second line is starting with 5. This is intercept. That means there is a systematic error. You are getting this difference in all measurements. This is called systematic constant error. But suppose if this difference is increasing with increasing concentration, either on downward side or upward side, this is called systematic proportional error. Error is increasing with increasing concentration. So ideally what should happen? Our line should be parallel, should be almost overlapping with this 45 degree line. Now we want objectivity in everything. How we can develop objectivity in this? Suppose you have to compare 3 different machines, B, C, D, with your A. 3 companies, they are approaching you. What you can do? You run one set of samples on machine A, then second set of samples on machine B, C, same set of samples on machine B, C, and D, and then draw this graph. A versus B, A versus C, and A versus D. Then see correlation, where you are getting best correlation for most of the parameters. And to bring objectivity in the system, what you can do, you can take the help of Westgard website, because this calculation is, formula is a very cumbersome formula. But on Westgard website, if you put in machine A, your readings which you have plotted on x-axis, then on machine B, readings which you have plotted on y-axis, and when you are submitting, then you get graph of this type and one R2 value, that is correlation coefficient. If it is more than 0.90, that means this is very good correlation. If it is more than 0.95, this is excellent correlation. This should be as close to 1 as possible. And if suppose you are starting a new laboratory, or you are starting some new parameters for which you don't have any machine, then for this A, for x-axis, you can take the help of some of your colleague. You send set of samples to that laboratory and BCD you're testing on different machines, then you can do the same comparison.
Sir, I think your voice is not reaching us now.
Yeah, actually, there is this problem in the slide. The slide is not moving forward.
Okay. Maybe you can escape it, sir. Well, maybe you can just try escaping it once and then redoing it.
Okay, are you trying?
I cannot, sir. Only you can.
Okay.
So you can move away from the slideshow mode, go to the proper mode, and then come back to slideshow mode. So it should be okay.
So you can probably try escape on the computer. It will let you out of this presentation mode. So you can also right click on the screen and say end show. Do a right click on the screen and say end show.
I think your system has stuck somewhere. So Priyanka, I think should sir stop sharing for now and then start sharing again?
Yeah.
Yes.
I think that will be better.
You can take the screen as of now.
No, now it is.
Yeah, it's okay. It's okay. It's okay.
It's okay.
Thank you.
Now, after this, I have always seen a lot of confusion regarding understanding of this term. Validation, verification, and sometimes calibration. Although we use these terms frequently in our laboratory, sometimes people say we have done validation and verification both. And if suppose somebody is saying I have done validation and verification both, that means there is some confusion regarding the understanding of these terms. Validation and verification. What is validation? Validation, if we go by the definition of the books, confirmation through provision of objective evidence that requirement for the specific intended use have been fulfilled. Or if we see definition of verification, confirmation through provision of objective evidence that specified requirements have been fulfilled. What is the purpose of a definition? This shall clarify our doubt regarding the understanding of that term. But with this definition, I think all of you will agree with me, confusion has increased rather than settled down. In simple terms, what is validation? If suppose somebody is claiming something based on some research work, some study, that is called validation study. And that is some evidence, supportive evidence, whatever they have done, supportive evidence in support of their claim. If suppose some company person is coming to us, they are saying, in our machine, you will get throughput of 100 tests per hour, precision of 2%, and linearity up to 25 gram per dl for hemoglobin. How they are making that claim? Based on some extensive work in their R&D lab. Then generally they take the help of good laboratories, some government organizations, some private hospitals, some standalone laboratories, different type of small, large, different type of laboratories where they are planning to install those machines, and then they come to a user. We have developed this machine and we have checked in our R&D, and various other, these users, they have also checked and provided us this certificate. These are the supportive evidence in support of their claim. That is called validation. Generally, we say validation is at the level of manufacturer and verification is at the level of user. They're the same thing. And when we tell them, okay, you are saying, whatever you're saying, that is okay. But we want to check this machine in our laboratory first. Then we will decide. Then they keep one demonstration model in our laboratory. When we check the same thing, linearity, precision, throughput, we are not claiming anything. We are just confirming their claim. This is called verification at our level. If suppose we are developing a new test, or we are using a method or machine outside its intended scope. If suppose you're buying a kit for serum calcium and you're saying, I want to perform urine calcium also with this kit, and manufacturer has no such claim, in that case, you have to validate because manufacturer has no such claim. Then if you're doing something, you're running control, you are comparing result with the kit design for urinary calcium, then this is validation at your level. So these terms should be clear in our mind, and generally I explain with a very simple story. All of you know the story of Shabri and Ramchandraji. Shabri offered bear to Ramchandraji, but before offering those bear, that fruit, she tasted that fruit. What was the purpose of her tasting? Whether this is good for eating or not. I am offering this to my God. Whether this is good for eating or not, it is my duty to check. And there is no other way of checking. And suppose, if, just assume a particular situation. Lakshmanji was sitting next to Ramchandraji. If he would have thought, I am his real brother, and she is outside party. Shabri is outside party. And when I am accompanying him, it is my duty to check whatever she is offering, whether that is really good for eating or not. अगर लक्ष्मण जी टेस्ट करते तो क्या होता? वो शबरी के क्लेम को कंफर्म करते. Whatever Shabri did, validation, and whatever Lakshmanji would have done, verification. But remember one thing, because I don't know how many of you have read Ramayana. In actual Ramayana, Lakshmanji didn't taste that fruit. Remember one thing, this is only to explain you validation and verification. Whatever Shabri did, validation, or you can say that is sort of validation, and whatever Lakshmanji would have done, verification. Now, what is calibration? What is our expectation with a particular instrument? We expect a desired performance by a machine. What is our desire? A pipette should measure correct volume. A thermometer should measure correct temperature. A centrifuge shall rotate at a correct speed, and for different analyzers, whatever results they are providing, those should represent actual value of the patient. This is our desire with a machine. So all these measurements should be correct. How we can have confidence on the measurement? When we are getting that checked by authorized people. Who are authorized people? All these non-analytical instruments shall be calibrated by a calibration lab, which is accredited as per 17025 for the calibration of that particular equipment. And that calibration laboratory shall have accreditation certificate by a authorized accreditation body. And these analyzers shall be calibrated by manufacturer or authorized distributor. And your analyzers, whether it is hematology analyzer, biochemistry analyzer, where you are getting quantitative results, these shall be calibrated with the calibrator only. Sometimes some people say, we are comparing result with a high-end model. No, that is not acceptable. It should be calibrated with the calibrator, and that calibrator is a sort of reagent where these analytes have a defined fixed value. And these calibrators shall have traceability to national or international standard. This is called metrological traceability. This process by which our results are traceable to national and international standard through unbroken chain of comparison is called metrological traceability. Now, after discussing all these terms, when we are evaluating a particular machine, we have to evaluate. This is important from company perspective also. If you are installing a particular machine, always check where exactly they are going to put that machine, whether that space is adequate and appropriate for that machine or not. And this is important from user perspective also. You discuss with company people, we want to keep this machine here, environmental condition and other conditions, whether these are acceptable for this machine or not, and what kind of service you will get from the company people. What is the extent of technical training required to use that machine, and usefulness of instruction manual, whether it is written in a very complicated language or in a simple language, whether your people are able to understand that or not, or whether company people can simplify that for your people or not. Assessment of safety, then linearity also. What is linearity? Maximum value of an analyte which you can report without dilution is called linearity, and the maximum value which you can report with dilution is called reportable interval. Because minimum value will remain the same, the difference between minimum value and the maximum value, that is your reportable interval. Precision. What is precision? You are getting similar result on repeated measurements. Carryover. What is carryover? After running a analyte of low value, you are running a sample with high analytical value. After that, you are running that low value again. If your second result of low value is affected by your higher result, then this is called carryover. You have to see machine shall not have significant carryover which is affecting the decision taken based on that result. Then comparability with an established method. And if you are participating in EQUAS, what is the performance of that machine in the EQUAS? Previously, we discussed diagnostic sensitivity, diagnostic specificity, positive predictive value, negative predictive value. Now we are talking about analytical sensitivity. What is this? This is the minimum difference which you can get in analytical measurement in a particular machine. Suppose there are three machines. One is measuring hemoglobin 10, 11, 12, 13, 14, 15, like that. They are detecting a difference of 1. Another machine 13.1, 13.2, 13.3, they are detecting a difference of 0.1. Another machine 13.11, 13.12, 13.13. This machine can detect sensitivity, detect a difference of 0.01. This minimum difference which a machine can detect is called analytical sensitivity. In first case, this is 1. In second case, it is 0.1. And in third case, it is 0.01. You have to decide based on your requirement which is best suitable for your laboratory. And analytical specificity means when you are measuring, your reagent system is detecting only that analyte. In addition, it is not detecting anything else. Sometimes what happens when you develop reagent system for a particular analyte, suppose it is detecting some other thing which is present in very small quantity in that specimen. Your result is not affected by that. But if that is present in significant quantity, then quality of a result is affected by that. So you have to see analytical specificity also. And how much time a machine will take in analyzing single specimen. Then you check throughput time. Suppose you have to provide 3-4 wash cycle after analyzing 20 specimen, or you have to shut your machine for half an hour or 1 hour after analyzing 40 sample or 30 sample. This will decrease your throughput time. Your analytical time may be less. And reliability of your instrument when in routine use, and adequacy of service and maintenance provided. Suppose there is frequent breakdown, you have to call your company engineer again and again for various things, then reliability will be doubtful. Cost per test. Sometimes we think, okay, in this machine, our reagent cost is 15 rupees per test. And we are charging from the patient 100 rupees, it is okay. But remember one thing, in this cost per test, you include operating time, other reagents, cost of control, cost of calibrator, and also include cost of your CMC or AMC, and also include cost of your machine. Suppose life of machine is 5 years, and you're taking AMC, then you see how much cost you can attribute to machine cost, how much cost you can attribute to CMC, how much cost you can attribute to calibrator control. Then you decide cost per test. That is your actual cost of running a particular sample. And also include that washing cycles, staff acceptability, level of technical expertise required, any relevant authority level like CE level, etc., if it is required. Now you're using machine. Machine will give you good performance if it is being maintained properly in the lab. What is required? You have to see what is the manufacturer recommendation for daily maintenance, for weekly maintenance, for monthly maintenance, and periodically you are getting preventive maintenance by the manufacturer. You should have proper AMC or CMC from the manufacturer for the proper servicing of your machine. Now we have discussed so many things related to machine. We have to maintain records also, because whatever is not recorded and whatever record you are not able to produce, people will assume you have not performed that activity. What are these records in relation to your machines? Sometimes this people say, they ask company people also, what is this IQ, OQ, and PQ? We are buying your machine. You organize whatever records you are providing to us in this IQ, OQ, and PQ manner. Create different files for this IQ, OQ, PQ, or in the same file you separate a sheet. What all are included in these IQ, OQ, and PQ? IQ means installation qualification, OQ means operational qualification, and PQ means performance qualification. But before all these things, this is pre-installation. Some documentation related to site assessment, important for both party, company as well as laboratory people. And here remember one thing, I always tell in my various lectures, the relationship between our industry partners and laboratory people is like body and soul. Both cannot exist without each other. And who is body and who is soul? That is again a question mark. But one thing is certain, and I think all of you will agree with me, both these parties cannot exist without each other. So that we should develop a system we are learning from each other. In pre-installation site assessment, good for everybody. Company people are happy. We have checked that they are going to install machine and their space is okay. Laboratory people are also happy. Then copy of purchase order, copy of invoice, a parts list, operational manual, procedure of equipment, and SOP of various tests, training package. All these things should be there with the machine. Now we are starting handling our machine. When we are opening pack and keeping machine on the shelf or on the floor, examine general appearance of the machine. You have bought a new machine, okay, it looks like a new machine. There is no damage to machine during transportation, and it looks like new machine, not an old machine or used machine or refurbished machine. And serial number on the machine is matching with the serial number on various documents. Then how they are assembling various components and how they are assembling reagent system in the machine. What all environmental conditions, whether these are suitable as per machine or not, power check, training and post-training evaluation documentation, support service, name and contact detail of the engineer, because till this time we are interacting with the service people. Now from here we have to interact with engineers also, support people. So name and contact detail of that. And in addition to that, service engineer, these companies, they also have product specialists, people with technical knowledge of various parameters. So you should have contact number of those people also. So we should have details of our marketing people, sale people, product specialist, as well as service engineer. Then these IQ documents are reviewed and approved by both parties, laboratory as well as suppliers. Then calibration schedule and procedure, actual performance of calibration and instrument printout related to that. Also keep purchase vouchers of that calibrator with you, maintenance schedule and procedure. Then linearity, that linear regression, precision, carryover, documentation of all these things in your laboratory. And whatever you are getting, these are called performance characteristics. Document that, keep instrument printout also in your record. Check with controls, keep that also in record. Check with tested samples, what were the results with the previous, with the other machine, what are the results with this machine. After all these things, this operational qualification is reviewed and approved by authorized person. Now in performance, maintenance, if suppose frequent repair is required, you are calling company engineers frequently, then you will have some lack of confidence about the maintenance, but keep all these things, maintenance record, performance in IQC and EQUAS, who are your key operators of the machine. Then this PQ is also reviewed and approved by both parties, and then final approval signature. We have checked everything, now this machine is performing well, and we can use this for patient testing. In addition, in our laboratory, we shall have safety protocol in relation to physical hazard, fire safety, electric safety, many other things, chemical safety, biohazard, and disposal of biomedical waste, which should be as per your national or regional biomedical waste guidelines. Sometimes some people say, we are participating in IQC. Therefore, what is the need of EQUAS for us? It is not required by us. But remember one thing, with IQC, we are checking precision, and with EQUAS, we are checking accuracy. Precision is we are getting similar result on repeated measurement, and accuracy is how close our result is to true value. And the difference between your result and the true value is called bias. This difference is called bias, and higher the bias, less is the accuracy. And for this precision, what we do, we check standard deviation. Higher the standard deviation, higher is the variation, and less the precision. One thing is certain, we need both IQC and EQUAS. How many controls are required, how frequently these shall be run in the laboratory, this is based on workload and distribution of your work, and what type of samples you are getting and what type of quality you are looking at. In this IQC, laboratory shall have documented procedure, then implementation of that procedure, then a system of reviewing that data generated, and when there is any outlier, some acceptability criteria, you're detecting that outlier, you're taking corrective action, and you're monitoring that corrective action for its effectiveness. And all this documentation is signed by some approved person, or you can say authorized person, who is releasing the patient reports. And how these are evaluated, how you're detecting outlier, we use these LJ charts and Westgard rules, and we can use Six Sigma also. This is complete lecture in itself. What are the various reasons for outliers in IQC, and how to interpret these LJ charts? What is Six Sigma? These things we will discuss in separate sessions. In hematology, we cannot use these LJ charts directly because stability of the controls is less. Sometimes stability is only 1 month. Whatever mean and SD we are generating with the previous lot, that cannot be used by our control of next month because there is change in the lot. What we can do in that, we can calculate CV percent for different lot, and we can compare that CV percent. If suppose CV for lot A is 2%, CV for lot B is 2.1%, CV for lot C is 2.3%. The CVs are comparable, that means our precision is okay. But suppose for last 6 months, CV was close to 2, and in one particular month, CV is coming 4%. That means something has happened. That CV is not acceptable. Sometimes people ask, what is acceptable CV? how we will decide whether the CV is acceptable or not? Then remember one thing. With time, your CV should decrease. Same system cannot be applied to all laboratories. If suppose in a particular laboratory, they are starting their quality management system and suppose their CV is 6%, say for glucose. Next month their CV is 5.3, then their CV is 4.8, then their CV is 4.3, and then their CV is 4.0. What will it say? Their CV is improving. That means their quality is improving. Lesser the CV, better is the situation. But suppose in a lab, their CV is close to 2, 2.1, 2.2, and one month, particular month, they are getting CV 4%. Then what will you say? There is some deterioration in the quality. So you cannot apply this 4% to both laboratories. Our intention should be keep this CV as minimum as possible. And in hematology, for calculation of this CV percent, use absolute value rather than percentage of DLC, because percentage, and then you are calculating percentage of that percentage, then mathematically that is not very meaningful. For that CV percent, when you are using this hematology analyzers, use absolute values. And how we assess this EQUAS? For quantitative terms, we use Z-score. For qualitative, we use comparison with peer group or evaluation criteria. What I mean to say, suppose you are participating in EQUAS for AFB in sputum sample. International norm is there should be more than 75% concordance in the test. Results should be similar in more than 95% of participants. If suppose in 10% participant, they are saying result is negative, and 90% they are saying result is positive. These 10%, that means these 10% have not submitted correct result. Their result is not correct. But suppose 40% are saying result is negative, 60% are saying result is positive, then we have to discard that cycle. We cannot decide on the basis because there is some issue related to quality of the specimen. The specimen is not a representative sample of that particular EQUAS material. So that thing we have to keep in mind, and these things, these PT providers also consider. Now, we have discussed so many things. We have discussed our pre-examination part also. If there is any deficiency in the pre-examination or any other thing, relevant point related to report, that shall be communicated to the clinician. Suppose there was some problem related to labeling, sample was not labeled when you received that, and that was very precious sample, it was CSF. It is not possible to collect again, and immediately one pediatrician came to you, I forgot to label that in hurry. This belongs to patient so and so, and I'm labeling in front of you, and I'm signing on it also. Then this information shall go with the report. If the sample was unlabeled, however, the doctor identified that sample and labeled himself in my presence. Now, our result is ready. We have checked quality also in that sample. We have to review patient result with the previous results. This is called delta check. So that we are sure there is no sample mix-up and no problem related to identification of the patient. Correction for interfering substance. Clinical correlation is also very important. And you have to check in hematology your histogram, your scattergram. Also see how this numerical data is correlating with each other. If suppose your hematocrit is, normally hematocrit is 3 times of hemoglobin, but suppose hemoglobin value is high, hematocrit is low, MCV is very high, MCH is also very high. That means these findings are indicating some cold agglutinin disease. Similarly, there are other things also. One or two examples we will discuss in this presentation, but there are many examples. Some flags are there. What is the meaning of these flags? If suppose some spurious results, very high hemoglobin and TLC, you may find these in lipemic sample. You have to keep in mind, rather than diagnosing some serious disorder. After reviewing patient result, when you're posting these results, you have to see there is no typographic error. And only authorized people are able to access that information. Whether it is hard copy of your result or you are uploading result on your website. Maintain patient records properly for a defined time and maintain in such a way the records are easily retrievable and retrievable in a legible manner. Monitor turnaround time, and whenever you think you are not able to turn around time for any reason, either because you ask for repeat sample or instrument was down, that shall be communicated to affected party immediately. And also you should have some system of taking customer satisfaction surveys, reviews, and also review that data. Whatever you are getting in that feedback, in that survey, you review those periodically and take appropriate action on that. This is a very simple test being performed in every laboratory. Quality is important here also, and very important. How you are preparing a slide, it should be of good quality. Quality of the slide should be good, quality of the spreader should be good, and you have to take appropriate size of the blood drop. Then how you are staining. Before submitting slide to a pathologist, technician should see whether the staining quality is appropriate or not. And when you are examining slide, you should be able to recognize, perceive different morphological features. And these morphological features shall be interpreted correctly. Because sometimes what happens, suppose you are able to see ruler formation. And whenever we see ruler formation in aged patient, we start thinking in terms of multiple myeloma. But remember one thing, in multiple myeloma, you get ruler formation, and ruler formation is not the only feature of multiple myeloma, not a diagnostic feature of multiple myeloma. But in multiple myeloma, you get ruler formation in all directions. But if this is artifactual, you get ruler formation only in one direction. If you are finding target cells only in one part of the slide, one portion of the slide, that may be because of drying artifact. In pathological target cells, you find target cells all across the slide. So all these things, this is one example. Similarly, when you're interpreting various things, you have to interpret seriously. That is why we say performing a test is one thing, but this test shall be interpreted by a qualified person in the laboratory. This is must in every laboratory. And sometimes people say we are performing only some routine test and we are testing only 25, 30 samples or 40 samples in a day. Our laboratory is not a very big laboratory. Then what is the requirement? All these quality issues and accreditation. Remember one thing, if a patient is coming to you for a glucose, for him that glucose result is very important. He is not bothered what all tests you are performing. You are performing COVID-19 or histopathology, immunohistochemistry, microarray. That patient is not bothered. Patient is bothered about his or her glucose result. That should be, and whether you are testing 25 samples or 250 samples, he or she is not bothered. Like a 35-year female came for annual health checkup. There was difficulty in sampling. Tube was only half filled. You see here, normal parameters. Clinician thought this is a microcytic hypochromic picture with normal hemoglobin because MCV was 72. He asked for iron studies. It was normal. And then he was planning to go for HPLC for hemoglobinopathy because microcytosis with normal hemoglobin, he thought about hemoglobinopathy. But then he thought I should go for repeat CBC. And when repeat CBC were performed, it was normal. What was the reason? You see here, these are not correlating with each other. 13.5. This hematocrit should have been around 40. What was the cause of this microcytosis? It was only half-filled tube. Excess EDTA causes shrinkage of red cells, therefore decrease in the mean corpuscular volume. And in our analyzers, hematocrit is a calculated parameter. Because of this low MCV, this was low. The laboratory person should have checked this. There is some problem. And samples also shall be checked whether the samples are adequate and appropriate to perform a particular test or not. With that, we can avoid many problems for the patient, for the clinician, and for us also. You see, low hemoglobin, low TLC, platelet. This is a case of pancytopenia. But before releasing that result, technician noted a plot. This should have been noticed at the time of accepting sample only. Or when the test is ready, in some cases where it is required, you test sample again, whether there is some problem related to sample or it is okay. This case, high hemoglobin, high TLC, maybe because of dehydration. We have to keep in mind rather than thinking about some serious disorder. Again, a case of pancytopenia. In this, sample was collected from the same arm where the patient was receiving IV fluids. Otherwise, the patient was scheduled for bone marrow biopsy. And when repeat CBC was performed, it was normal. Therefore, they were able to avoid one unnecessary invasive procedure. And if suppose everything is okay, whatever data we are generating, that shall be interpreted properly. In this, clinician thought MCV is normal. This is normocytic normochromic anemia. Sometimes people ask for only few parameters. They don't ask for peripheral smear, and laboratory they perform test, whatever being asked for. So he thought this is normal MCV. But our analyzers, they give us average value. If suppose in a case there are microcytes as well as macrocytes, you may get normal MCV. When peripheral smear was examined, it was a case of dual deficiency. When this patient received hematinics, iron as well as B12 and folate, then her hemoglobin improved. Important thing is, if we see our today discussion with a little extra care in different phases in our laboratory, in pre-examination, in examination, in post-examination, and if we are very careful at the time of deciding our machine, at the time of deciding our personnel, and we are helping clinician in the interpretation of test and also in deciding about test, when we help them in deciding about the test, this is called pre-examination. And when we help them in interpretation of a particular test, that is called post examination. So all these are important, because with this little extra care, we can save one more life. We have various other webinar sessions. For these, you can contact either my secretary, Tripti, or me. One we have on risk management in laboratory on 17th. Whoever is interested, you can send us a WhatsApp message. We will send you link for that. These are the list of various programs in April and in the first half of May. And the mantra for good life is stay at home, maintain social distancing, stay safe, and when you can't do much, in addition to whatever you are doing, cooking, playing Ludo, everything, you can enhance your knowledge, which you can use in better care of the patient. And all these things, when you are at home, you are busy in all these things, you can keep your body and soul together. Thank you very much.
Thank you, Dr. Neeraj, sir. If you have allowed some time, there are 2-3 questions which I would like to ask on the behalf of participants.
Definitely.
Okay, sir. So the first question was regarding this lockdown period. Someone has asked that if control is not available in this lockdown period, what should be done? What should be the, how to ascertain that what I'm doing is correct?
If suppose you don't have control, what you can do, you can develop some acceptability criteria. Actually, you should be ready for any situation. This acceptability criteria you have to develop beforehand. You know, you have controls for only suppose 15 days. You otherwise also to see whether you are storing samples properly or not, you test some stored sample. So how much difference you are getting? Suppose for hemoglobin, in the morning it was 13.2 and in the evening it is 13.4. Difference is, or next day it is 13.4. Difference is 0.2. Similarly, a sample tested tomorrow, you are testing again day after tomorrow. Suppose for hemoglobin difference is 0.3. Someday difference is 0.1. That way you collect 30 data points. How much difference you are getting? And whatever difference you are getting, you take the mean value of that. One day you are getting 0.2, one day you are getting 0.1, 0.3, 0.2, 0.1. These type of differences you are getting for hemoglobin. And at the end, you calculate mean and SD. Suppose your mean is 0.2 and your SD is 0.1. So mean plus 2 SD, 0.2 plus 0.2, 0.4. So you can say, based on my data, my acceptability criteria is if a hemoglobin variation in interday hemoglobin variation is more than 0.4, I will not accept that result. If suppose on a particular specimen you are getting difference of more than 0.4, it may be because of some stability in the poor stability in the sample. You test one more sample or two more sample. If again this difference is high, that means something has happened, because in this short time nothing happens to generally reagent. What happens, suppose there is some leakage or blockage in the tube, that you will be able to detect. You can use stored sample in that situation. Next question.
Right.
Thanks a lot for the informative answer. Sir, the next question is regarding IQ, OQ, PQ frequency, should it be done every year or only when the
No,
instrument gets?
no. IQ to it is installation qualification. When
Okay.
you are purchasing a equipment, operational qualification, whenever you are appointing a new person who is not trained, you arrange training for that person to operate that machine. That training may be through company person, or some already trained person can also train a new person. This precision, linearity and carryover, that shall be checked once in 12 months.
Okay.
And performance qualification, that is ongoing process. You are participating in IQC, IQAS, so that is an ongoing process.
Sir, in furtherance to the first question, can the retained sample act as a QC for the time being as long as QC
yes,
is not
this is
available?
what I said. You can use retained sample as IQC for the time being, and you get one letter from the company people ki in this time controls are not available. So that your intention is clear, you want to buy, but those controls are not available. But for retained sample, you shall have some acceptability criteria.
All right, sir.
How
Understood.
you accept reagent. So these acceptability criteria you have to develop for your hemoglobin, for your TLC, for your platelet, for your RBC. I'm talking in relation to hematology. For your biochemistry, you have to develop acceptability criteria for different parameters. Because sometimes what people say, we are following clear guidelines. They are saying 20% variation is possible. They are saying 50%, 15% variation is possible, is acceptable, without realizing the fact that what is the meaning of 20%? 20% means if suppose I am saying hemoglobin is 12 today, and tomorrow I will accept 20% variation. That means 20% variation means 2.4. If suppose my hemoglobin is 9.6, I'm accepting that. If my hemoglobin is 14.4, that I'm also expecting. Is that reasonable? You have to develop your acceptability criteria.
Okay, sir. One more question, sir. In hematology, what should be the calibration frequency for the instruments?
Actually, what manufacturer they recommend, they say once in 12 months.
Okay.
Generally. And what accreditation body is saying, they are also saying once in 12 months, these analyzers. But particularly in relation to hematology, what most of the big institutions who have very high workload, they say these equipment shall be calibrated once in 6 months. So that means ideal situation is once in 6 months. But if your workload is reasonable, and if you're going by the manufacturer and by the accreditation body, you should get your equipment calibrated. Or even if you're not going for accreditation, you get your equipment calibrated at least once in 12 months. This I'm telling you in relation to hematology. But in biochemistry, various parameters, they go out of calibration more frequently. So you have to get that calibrated in your laboratory only by calibrator. These hematology analyzers, these are calibrated by company people only for this once in 12 months. And otherwise also these biochemistry analyzers, hormones analyzer, etc. also shall be calibrated once in 12 months by the manufacturer.
Sir, one last question. These record of maintenance, calibration, QA, how long should be retained in the laboratory?
As per ISO 15189, clause 5.3.1.7, this should be maintained for the whole life of the instrument.
Life of the instrument.
Not IQC and IQAS. This related to instrument maintenance and purchase for the whole life of the instrument. IQC and IQAS record, if suppose you are going for accreditation, you have to follow recommendations of the accreditation body, and otherwise maintain these records till the next accreditation, or at least 3 years.
All right, sir. Thank you. With your permission, one more question I would like to ask,
Sure.
if you allow. Okay. So let's say a laboratory is running 3-level QC every day for hematology. And one fine day, they find that one level has not passed the criteria. So to fulfill that deficit, can they run the retained sample if they have already established the criteria?
No, they have to first understand what is the reason. If suppose their normal control and high control, these are okay, but there is some problem with the low level control. Problem may be in the control material itself. They should run with the new control, new vial of the control. If suppose they don't have new vial of control, then what they can do, they can run a sample with low value. If suppose they are facing problem in the hemoglobin, in the lower range, then they can run a hemoglobin of lower range. If suppose in the morning it was 6.2, and then now it is 6.4, but there was variation in the control material, they can say we tested with the control material, we tested with the patient sample of low result, and with that we are approving this. But we have to ensure quality of the samples of low range. Next. Hello.
Hello, sir. I think
Thanks a lot. We are done with the question and answer. Over to Priyank.
so. Thank you, sir. Thank you again for taking out your valuable time for sharing these insights. I think they can, really be of use in day-to-day lab practices. So thank you for your time, sir, and thank you for sharing your expertise. We are indebted to you for your knowledge and kind sessions. So may I, for the people who are attending the session, I would like to remind you that the whole session, the recorded session, will be available on Mindray India Facebook, YouTube, LinkedIn pages. So at your disposal, you can always, use this as a reference at a later stage as well. So thank you again for joining with us in this conference, in this masterclass. And we look forward to see you again in some webinar that we are conducting.
Thank you very much, everyone, for sparing time. And thank you, Mindray, for inviting me to conduct this session in your forum. Thank you very much.
Thanks a lot, sir. Have a good time.
Good day, sir. Good day to everybody. Thank you.